This is Dr. Jeremy Graff, Chief Scientific Officer and Senior Vice President for Research at Biothera Pharmaceuticals. In this module, I'll describe for you Imprime PGG and a mechanism-based patient selection marker that we use to identify patients most likely to respond to Imprime-based therapy. Let's review briefly how Imprime works. Therapeutically, Imprime works by activating multiple points of the anti-cancer immunity cycle. It activates tumor cell killing. It enables the repolarization of the immune microenvironment, so it's now permissive for immune eradication of the tumor, and it activates effective antigen presentation capacity. Ultimately, these events culminate in the activation of T-cell-based anti-cancer immunity, allowing for T-cells to become activated. These activated T-cells can then be unleashed by the checkpoint inhibitor therapies. So what is Imprime? Imprime is a beta-1316 glucan isolated from the cell wall of the yeast Saccharomyces. It acts as a pathogen-associated molecular patterning molecule, or a PAMP. PAMPs trigger the activation of the immune system. Imprime is the only systemically administered PAMP. When it's injected into the bloodstream of patients, Imprime is immediately bound by anti-beta-glucan antibodies. That immune complex then activates complement and becomes decorated by complement opsinin. This three-part complex is represented in the cartoon at the top. Importantly, Imprime responsiveness requires formation of this complex. If a patient has insufficient levels of antibody, the patient does not form enough complex and therefore does not respond to Imprime-based therapeutics. Because Imprime acts as an immune complex, the formation of the immune complex then allows us to understand who would be most responsive to Imprime. We've done this through a series of studies. First, ex vivo human donor studies, where we've taken blood from volunteers and we've asked whether or not the activation of innate immune cells would be related to levels of antibody in these donors. We've also looked retrospectively at our randomized clinical trial data to ask whether or not higher levels of anti-beta-glucan antibodies might mark those patients that benefited most from Imprime. And lastly, we've done healthy human volunteer clinical studies to establish the linkage between anti-beta-glucan antibodies and the pharmacodynamic effects that are elicited by Imprime upon injection. Let's focus first on our ex vivo studies. We know different donors have different levels of anti-beta-glucan antibodies. In this case, we've taken a donor who has insufficient anti-beta-glucan antibodies who does not respond to Imprime. You can see this in the context of both IL-8 and MCP-1 expression in this patient. When we supplement this patient's blood with ever-increasing levels of the antibody, with anti-beta-glucan antibodies, we restore the expression of these important chemokines and cytokines. Similarly, we can look at the function of innate immune effector cells. In this case, we're looking at the generation of reactive oxygen species by neutrophils. In this donor, without supplementation with ABA, there is no generation of reactive oxygen species. When we add a little ABA and then we add more ABA, we can now rescue the generation of these reactive oxygen species. These types of ex vivo data show us the importance of anti-beta-glucan antibodies for innate immune activation. We can also look retrospectively at our clinical trial data. In this particular case, we're looking at the trial that was run with Imprime and Erbitux in third-line colorectal cancer. We're looking at both progression-free survival and overall survival in patients treated with this combination. You can see for both clinical endpoints, those patients who had higher levels of anti-beta-glucan antibodies benefited more profoundly than those patients who did not. Importantly, when we looked at the control arm, those treated only with cetuximab, neither PFS nor OS was different based upon the levels of these anti-beta-glucan antibodies. These data collectively then tell us that only in Imprime-treated patients do we see benefit related to levels of anti-beta-glucan antibody. We next evaluated whether the immunopharmacodynamic effects that happen immediately after the administration of Imprime might be linked to antibody levels, to anti-beta-glucan antibody levels. In this particular spiderweb plot, you can see on each spoke a different chemokine or cytokine. The central core represents no change in gene expression, and every concentric ring thereafter represents an increase by an order of magnitude in expression of these chemokines and cytokines. We've looked at patients who are positive or have sufficient levels of the anti-beta-glucan antibody, and two different types of what we would consider to be biomarker negative patients, those who have insufficient levels of the antibody or those who only have an IgM isotype of anti-beta-glucan antibody. Those biomarker negative patients are marked in black and red, and you can see they do not respond to Imprime. Conversely, the patients who have sufficient levels of the IgG anti-beta-glucan antibody are marked in blue. They show us increased expression of these different chemokines and cytokines. They show us the pharmacodynamic effects. These type of data then help us solidify our understanding for the necessity of anti-beta-glucan antibodies. These data suggest to us that patients with anti-beta-glucan antibodies beyond 20 micrograms per ml might be our best responders. Those with antibody levels below 20 micrograms per ml would not be responders. So we next asked what the distribution of ABA might be in different cancer populations. You can see three different cancer populations represented here: lung, breast, and ovarian cancer populations. You can also see the three different Biothera trials, one in colon and two in non-small cell lung cancer. In every case, the median for these populations is right around 20 microgram per ml. We've chosen for this phase 2 setting to use 20 microgram per ml as our cut point. Those below 20 microgram per ml will not be included in our trials. Those above 20 microgram per ml will be included in our trials. What's represented here are the roster of phase 2 clinical studies ongoing with Imprime and pembrolizumab. The first study in non-small cell lung cancer is an investigator-initiated trial. This study is not using the patient selection biomarker. The other three studies marked in green, metastatic melanoma, triple-negative breast cancer, and head and neck cancer, are all using this patient selection criteria to identify the patients most likely to respond to Imprime. For additional information, please visit our website. Thank you for your attention.